Thanks to the organizers for having me here. It's my pleasure to present KNP301 on behalf of Kanaph Therapeutics. KNP301 is a novel bispecific fusion protein that targets both the complement as well as the VEGF components for the treatment of geographic atrophy as well as wet macular degeneration. I think we're all familiar with the pivotal role that anti-VEGF plays in wet AMD, but there's increasing evidence to show that complement is also involved. We find complement activation at the scene of the crime. Eyes with wet AMD have increased activation of complement, and this complement activation in the eye can drive microglia activation, neuroinflammation, that together lead to the secretion of inflammatory cytokines such as IL-6, as well as inflammatory VEGF production. So altogether, they can increase vascular exudation in the short term, and in the long term, this complement activation also leads to delayed complications of macular atrophy and fibrosis. So wouldn't it be nice if we have a single therapeutic that can suppress both VEGF and complement activation to increase short-term efficacy as well as to allay the long-term complications that reduces vision in wet AMD patients. On the geographic atrophy side, we are well familiar with the role of complement, but there's also evidence to show the involvement of VEGF. Current treatments, Syfovre, Izervay, unfortunately increase the new onset exudation in GA eyes, and this, when it comes about, can drive worsened outcomes such as decreased BCVA and increased lesion expansion. Fortunately, in these eyes, anti-VEGF treatment can reduce the exudation. But however, in this context, patients do face a very high treatment burden regimen in having separate concurrent anti-complement, anti-VEGF therapeutics. Again, it'd be nice if we have a single therapeutic that can come together to prevent rather than promote a new onset exudation in the eyes and GA. And for patients that already have both, this could be a two-in-one treatment for a more burdensome and more efficacious treatment for these patients. It's with these unmet needs in mind that Kanaph developed KNP301. On one end of the molecule, it binds C3b, the active moiety that gives rise to complement amplification. And on the end of the molecule is a aflibercept-like VEGF binding domain. So together, it is able to bind human C3b and human VEGF at very high affinities in nanomolar to picomolar affinities. In terms of the overall shape of the molecule, it's very similar to faricimab and bevacizumab in size. However, Kanaph has engineered the molecule to be longer in terms of its hydrodynamic radius, and this translates to a longer half-life within the vitreous. And you see this here on the left side in rabbit PK studies, but also in NHP PK studies, that the vitreal half-life in the pink bar is 1.5 to 2 times greater than the comparative agents for dry AMD as well as for wet AMD. The longer size of the molecule doesn't prevent it from distributing from the vitreous to the retina. We find that from the NHP PK studies that the drug can move into the retina at the same rate, to the same amount as other anti-VEGF agents. So in terms of its potency, it's a very effective VEGF blocker. On the left, it blocks in the in vitro assay VEGF signaling to the same extent as aflibercept and faricimab. And in vivo, in the laser CNV model, you can also see equivalent performance of the molecule relative to aflibercept in terms of suppressing CNV leakage and volume. On the complement side, it's able to bind the much less common species of C3b. It doesn't have to soak up all the C3 that's present in the eye, and this translates to greater potency. And on the head-to-head hemolysis in vitro study, you can see that 301 is more potent than Syfovre and Izervay to about 3.5 to 5-fold more potent. And in vivo, it also works the same way in the sodium iodide injury model. 301 is able to suppress sodium iodide-induced inflammation, and with that, complement-mediated degeneration. So to summarize, we think that 301 can be the best-in-class treatment for both wet AMD and GA. In wet AMD, it's able to have increased short-term efficacy in addressing 2 pathways that are important for exudation. It has a long durability of action through its long vitreal half-life, and can also have the potential of suppressing the long-term complications of macular atrophy and fibrosis. On the GA end, we think that it can also be a superior performance from SLC agents through greater complement inhibition, a longer durability of action, and decreasing, not increasing, the risk of neovascularization. And to the very substantial fraction of patients that have both GA and wet AMD in the same eye, it represents a 2-for-1 treatment covering both bases at the same time. So KMP301 is currently a IND-ready asset that's completed GLP tox studies, that's ready for clinical translation following GMP development. Thanks very much for your interest.